CRISPR-Cas9 can get super confusing super fast.
So just keep in mind, we tried to keep this as simple and as clear as possible.
CRISPR, what does that even mean?
Well, Caitlin, CRISPR stands for Clustered, Regularly, Interspaced, Short, Palindromic Repeats. So, palindromic repeat. What is it? A palindromic repeat is able to read the DNA or the RNA in a front to back as well as a back to front direction.
Okay, before we start to teach you about the mechanism of the CRISPR-Cas9 mechanism, we need to teach you what CRISPR even is and where it came from. So CRISPR is a mechanism that bacteria have, kind of like their immune system. It was discovered in E. coli. And so how it works is there's palindromic repeats, and so that's segments of DNA, like Madeline talked about, that are the same and they're interspaced. So what goes between those spaces? Well, in between the spaces are segments of DNA that are unique to each other. And later on in the sequence was discovered DNA that was CRISPR associated. And that's what Cas stands for, CRISPR associated, and those genes code for proteins that are helicases and nucleases. So helicases unravel DNA and nucleases cut DNA. So this is a cell, and this is an intruder, some viral DNA that the bacteria doesn't want to kill it. You have your CRISPR system down here. The cell recognizes this viral DNA, and it has a sequence. One of these unique sequences matches this virus. It's like our immune system, our adaptive immunity, where the bacteria remembers and has stored this information. So what it will do is it will encode for a Cas protein, it will make an RNA for that virus. That virus's DNA will feed in, and it will essentially disassemble it and kill it before an infection even occurs.
Alright, so switching gears, now I'm going to talk about a remarkable protein known as CRISPR-Cas9. So, Cas9 is a protein which naturally occurs in many bacteria as an immunity mechanism. The Cas9 protein acts like a highly specific pair of molecular scissors that can cut and edit any gene in any organism. Cas9 was discovered by Jennifer Doudna and Emmanuel Charpentier in Streptococcus pyogenes.
Okay, so this is a Cas9 protein, and what Streptococcus pyogenes does is it'll create two strands of RNA. One is called a CRISPR RNA, which codes for the sequence of the DNA of the viral intruder. Now, the second strand of RNA is called a tracer RNA, and it essentially holds the CRISPR RNA in place. Now, what Jennifer and Emmanuel discovered was that if we could fuse these two pieces of RNA and create one strand, we would have a really simple system that we could manipulate, and all we would need to know to target a gene is the sequence of that gene. So here we have the connected CRISPR-Cas9 system. So this fused piece of RNA, they decided to call the tracer RNA CRISPR RNA chimera. And we'll just call it the guide RNA, because that's what it does.
Just to clarify what we've been talking about, when we're talking about CRISPR-Cas9, as Caitlin had mentioned, CRISPR refers to the guide RNA. Cas9 refers to the protein that does the actual cutting of the DNA and the unraveling.
Now, the Cas9 protein has helicase, a nuclease, that can unravel and cut DNA. So what this protein will do with its assigned sequence of DNA is it will travel along the DNA of the cell until it finds a perfect match to this sequence, which we, the scientists, have put in and wanting it to target. So once it matches the perfect sequence, it will cut the DNA on both strands. Let me just draw what I'm talking about when I say the Cas9 travels along the DNA. Okay, so here we have CRISPR-Cas9 traveling along the DNA of the cell. Now what's happened here is the helicase of the Cas9 has opened the DNA by breaking the hydrogen bonds, and we have this bubble here. And what it's doing is it's traveling along and it's scanning the DNA for a perfect match to this CRISPR RNA that we've assigned it. So that RNA matches the sequence of the gene that we're targeting. And once it finds that gene, it will cut here and it will cut here, both strands of the DNA.
The first inactivates a gene. The second inserts a gene.
So let's talk about how CRISPR-Cas9 could be used on a gene that needs to be inactivated. Say you have a gene causing a disease. What you can use CRISPR-Cas9 for is to come along, you would put an RNA sequence of the gene that you're targeting on the Cas9. It would travel along like we discussed, find the exact location of that gene, and cut it. Now, what will happen, that's all CRISPR does. What will happen is the cell will try to come along and repair the gene. That results in a mutation, and the gene is therefore inactivated.
So, say we wanted a more complex way to insert a gene into our segment here. So, we would have three components to that. We would have our guide RNA, we would have our Cas9 RNA, like the previous inactivation of the gene, and however this time we would also have a new section of RNA that will then code for the new piece of DNA that's being inserted here. So this is our RNA piece here. We would then get it coded on the top. And just to clarify, this piece here, the cell will copy the RNA.